Hello, grade 11 students. Here we go with part 2 of the revision for semester 1. So, transgenesis, transcription, and translation. So, now let's start with transgenesis. What is transgenesis? Transgenesis is known to be the process by which a gene is transferred from one species to another, and this gene is expressed in the other species. Let's say, for example, I transferred a gene from a dog to a cat, and the gene of the dog was expressed in the cat. So this is called transgenesis. What the meaning of transgenic? Transgenic, it's an organism that carries and expresses a gene from another species. Let's say we said that transgenesis is the process where a gene is transferred from one species to another and expressed in the other species. So the mice, let's say we transfer a gene from human to mice. The mice, they carry the gene from human. So we call the mice, they are called transgenic mice. Why? Because they carry a gene that is from different species and they carry it. Okay? Let's move to talk about chromosomes. Chromosomes at metaphase, all of you know that they are made up of two sister chromatids. They are easily distinguished and they are aligned at the equator of the cell, forming what's known by equatorial plate. The substance that makes up the chromosomes often is called the chromatin. Chromatin is divided into DNA composed of 4 nucleotides. 4 nucleotides, they are called A, T, C, and G. A, T, C, and G, they are the nucleotides that form the DNA. So DNA is composed of 4 nucleotides, A, T, C, and G. All of you know that the DNA is double helix, is double stranded. So we have 2 stranded opposite to each other. So let's say this one of the stranded, we have these nucleotides A, T, G, C, A. This is the first strand. The second strand should be complementary for the first one. So A, the complementary for A, it's T, and the complementary of T, it's A. The complementary of G, it's C, and the complementary of C, it's G, and also the complementary of A, it's T. So you have to know that A and T, they are complementary. C and G, they are complementary, as well as G and C, T and A. So A and T nucleotides, they are complementary. So if we have in the DNA a nucleotide A in the first strand, the complementary for A, it's T, and the complementary for C, it's G. Every 3 nucleotides together, for example, in this case, ATG, they are 3 nucleotides. Either they are called triplet, or they have the other name which is called codon. So you have to take care about this. Every 3 nucleotides, they form what's known by a triplet, or they are called codon. I prefer to use the word codon, C-O-D-O-N, codon, C-O-D-O-N. So every 3 they code for a codon. So for example, if I told you the first codon, where's the first codon? This is the first codon, A, T, and G. This is the codon. So A, T, and G together they form the first codon. If I told you the first nucleotide, it's the A here. Okay, so you have to take care about this and you have to pay attention to this. So as I said, DNA is composed of nucleotides A, T, C, G, and every one of these, A is called a nucleotide, T is called a nucleotide, G is called a nucleotide, C is called a nucleotide, and A is called a nucleotide. Every 3 together, for example, ATG, these 3 together, they are called codon, or they are called triplet. A and T, they are complementary. C and G, they are called complementary also. Proteins, they are composed of histones and non-histones. So we're going to work about DNA, about the codon, about the nucleotide ATC. In the process of transcription, DNA undergoes the process of transcription, where they gave mRNA, messenger RNA. And mRNA, by the process of translation, they give polypeptide, which is protein. Okay, so DNA by transcription, they give mRNA, and mRNA by translation, they give polypeptide. So here, what is transcription? Transcription is the process where the mRNA, messenger RNA, ribonucleic acid, is produced based on the given sequence of DNA. Mainly the process of transcription, it occurs in the nucleus. What is the fate of the produced mRNA? The produced mRNA undergoes mutation, and then they are transported from the nucleus to the cytoplasm, where DNA is going to give mRNA by the help of the action of two enzymes, helicase and RNA polymerase. We're going to talk about them in the coming slides. So transcription is the process where mRNA is produced on the given sequence of DNA. Let's consider the sequence of DNA molecule shown here. We have this sequence. We have GCA, TGC, ATC, TGC, CAT, and TG. This is called the non-template. Pay attention here to the names. This strand is given the non-template. Maybe they will give you the strand is called template. So what's the difference? Non-template is called the non-transcribed. Okay, and it's called the coding. While the second strand is called the template, the transcribed, the non-coding. Okay, did you pay attention to this? Transcribed, template, TT, non-coding. Non-transcribed, non-template, it's the coding. Okay, so this strand, it's the non-template, meaning that it's the non-transcribed, it's the coding. Why the complementary strand, it's non-template, opposite is the template. Non-transcribed, the opposite is transcribed, and the non-template, the opposite is the template. So how can we write down the sequence of the complementary strand? I'm not going to solve the first questions. I'm going to solve according to my own. So first, write the complementary strand. What's the complementary of G? It's C. What's the complementary of C? It's G. What's the complementary of T? It's it's T. So here you have to pay attention. You have to write them one under the other, and they should be organized well in order to have the fully organized and fully correct sequence. Okay. Second, we have T, the complementary is A. G, the complementary is C, and C, the complementary is G. Here we have A, it's T, A, and G. The complementary of T, it's A. The complementary of G, it's C. The complementary of C, it's a G. The complementary of C, it's a G. A, the complementary is T, T, the complementary is A, and then we have T, the complementary is A, and G, the complementary is C. This is the complementary strand. They said write down the sequence of the mRNA. mRNA sequence. It's similar to the template strand. It's similar, to the coding strand, the non-template. It's similar to the coding. So it's similar to the one in the blue, to this. But instead of T, it's going to be replaced by U. So it's the same strand, but instead of T, it's going to be replaced by U. So let's move here. The same sequence, non-template. We said the non-template is called the non-transcribed, it's the coding. So GCA, we put it GCA, the same. TGC, instead of T, we put U. So in the mRNA, T is replaced by U. Okay? And ATC, A is the same. T is going to be replaced by U. C. If we want to continue this, T is going to be UGC, CAU, and UG. This is in the mRNA, the T is replaced by U. Here we worked on the non-template, which is the non-transcribed, which is the coding. Okay, so the coding or the non-transcribed or the non-template, the mRNA sequence is identical, is the same. Instead of T is replaced by U. Some of you ask me, can we use the template, which is the transcribed, which is the non-coding? Yes, but we have to do the complementary. Look here. C, the complementary, it's G. G, the complementary, it's C. T, the complementary, is A. A, the complementary, is T. But in the mRNA, there is no T. It should be U. So here U. C, it's a G. G, it's C. T, the complementary, is A. A, the complementary, is T. But it should be replaced by U. And then G, the complementary, is C. And so on. So the third part, but how can you relate the complementary template strand to the non-template to the mRNA? Template is the transcribed, is the non-coding. So it's complementary, but instead of T, it's U. While the mRNA strand, it's the same as the non-template, but T is replaced by U. And this is the case here. Non-transcribed, non-template, the coding is the same, but T is replaced by U. To produce mRNA, helicase enzyme. Helicase unwind the DNA. It unwind the DNA of the gene coding for the protein. RNA polymerase will synthesize the complementary strand. For the transcribed strand will have the synthesis of the complementary. So this is the function of RNA polymerase and the helicase enzymes. Translation, we said the first process, DNA will give mRNA by the process of transcription, while mRNA will give protein or polypeptide by the action of the process of translation. It's the process by which an RNA, ribonucleic acid in specific mRNA, codes for production of proteins. Some of these proteins will remain in the cell, others are exposed. So given the following coding DNA strand, AAA, CCC, GGG, CAA, TCG. Give the sequence of the transcribed. It's complementary, where AAA, complementary is CTT, GGG, CCC, GTT, AGC. Specify the sequence. We specify the sequence of mRNA. It's the same as the coding. Instead of T is replaced by U. And we have to give the sequence of amino acid based on the sequence, based on the genetic code. So here, the genetic code table, where AAA here will give for protein lysine, CCC, every codon, every three will give an amino acid. Okay, GGG, CAA, UCG. Sure, you don't have to study them. They are given in the table. So this table, for example, UAA. Okay, the first letter is U. Here, the first letter is U. Second letter is A. For example, UAA, UAG, and UGA, you have to study them. They are called stop codons. For example, the first letter is A, the second letter is C. The first letter is A is here. The second letter is C is here. So THR. Okay, and so on. So this is the genetic code table. We'll start with the first letter and the second letter. First letter is here. Second letter is in the row. And the third letter, it's okay. So you have to study only three. UAA, UAG, and UGA, they are stop codon. Whenever we reach this, we have to stop because it's called the stop codon. And we're going to talk about this in details in the next presentation. So be ready. Thank you. Bye-bye. Now let's move for mutations. What's the meaning of mutation? What are the different types? And what are the effects of mutations? What is a mutation? Mutations, they are changes in the nucleotide sequence of the DNA. The term mutation refers to permanent changes in the DNA sequence. Mutations in the germ cells, they are transmitted to the next progeny, to the next generation, and may give rise to inherited diseases. Whereas mutations in the somatic cells, they are not transmitted to the next progeny, and they are important in the causation of cancers and some congenital malfunctions. The agent that can cause mutation is referred to as mutagen, like X-ray, gamma ray, UV ray, all of them, they are causes of mutation, so they are called mutagen. Mutations, they are divided either into frame shift mutation or point mutation. Frame shift mutation, like deletion, insertion, while point mutation can be silent mutation, missense, or nonsense. Let's Point mutation occurs when only one base in the DNA is altered. If it can be either silent mutation, there is no change in the sequence. Missense can change the sequence of the amino acid. Nonsense can lead to the presence of the nonsense codon, or is called the stop codon. There is no detectable effects in the silent mutation. There is no change in the amino acid sequence. For example, change from CGA to CGG, where A is substituted by G, does not affect the protein sequence since both of them they code for arginine. Missense, there is a different amino acid incorporated. For example, here glutamic acid is changed into valine GAA when A is replaced by U, or A is replaced by U here, and the G is replaced by G, and A is replaced by U. So here in the case of the normal blood cells, we are differentiated and developed to give sickle cell red blood cells. This is one of these mutations, the missense mutation. Nonsense mutation. It leads to the conversion of an amino acid into a stop codon. So nonsense, no way to continue. One type of thalassemia where the codon 17 of the beta chain is changed from UGG to UGA. G is substituted by A, and this results in the conversion of a codon tryptophan to stop codon, which is called the nonsense codon. Frame shift mutation, these occur when one or more base pairs they are inserted or deleted from the DNA respectively, either causing insertion if it's insertion or deletion if we delete one of the base pairs. For example, this is called deletion. So we deleted the G, so the sequence becomes ATG, then AAG. So we shifted the sequence of DNA to the left. This is called frame shift mutation by deletion. Or frame shift mutations, they have higher effects. Okay, since for example in substitution we change one of these letters. So the cat saw the dog. For example, we substitute the C with B. So the bat saw the dog. Still have a meaning. And the cat saw the hug. We substitute the D with the H. Or the cat sat the dog. Instead of W, we substitute it by T in the sat. So this point mutation has low effect, while the addition or deletion of a single base has much more profound sequences. For example, the deletion of K here, we deleted the K, so it becomes the ats, out, head, ug. So loss of C causes the destruction of whole meaning, and the gain of one letter. For example, I put M here between the C and A, so the CMA, TSA, WTH, EDOG insertion. So this causes the loss of meaning. This explains that the addition or deletion has a more and much profound effect or consequence on the sequence of the DNA. Okay?